Imagine unlocking the secrets of milk production at the cellular level, down to the very cells that make it possible. Chinese scientists have done just that, creating an unprecedented single-cell atlas of dairy cattle that could revolutionize how we breed cows and understand human biology. But here's where it gets controversial. Could these findings be used to create super-efficient dairy cows, potentially altering the natural balance of livestock genetics? Let's dive into the details. Led by researchers from China Agricultural University and supported by a global network of institutions, this groundbreaking study maps out the genetic blueprints of dairy cattle at an astonishingly detailed scale. Published in Nature Genetics, the research goes beyond traditional methods that only analyze gene activity at the tissue level. As Professor Sun Dongxiao explains, previous studies couldn't fully explain how different cell types work together to control traits like milk yield, fertility, or disease resistance. By zooming in to the single-cell level, the team uncovered interactions that were previously invisible to science. Using cutting-edge RNA sequencing technology, the researchers examined 59 different tissue types from Holstein cattle. They identified nearly 1.8 million high-quality cells and cataloged 131 distinct cell types, ranging from immune cells to reproductive cells. This is like creating a detailed map of a city, where each building, cell type, has its own unique function and neighbors. The team even discovered surprising connections. Excitatory neurons in the brain are linked to milk fat composition, while retinal cells and muscle cells also play roles in shaping the nutritional profile of dairy. And this is the part most people miss. The study didn't stop at milk production. It also decoded the complex cellular makeup of a cow's four-chambered stomach, revealing striking similarities to human stomach cells. This could open doors for comparative studies on digestion and disease. Additionally, the researchers classified mammary gland cells into 8 subtypes, with some directly tied to milk biosynthesis. Think of it as identifying the master builders in a factory, cells that are critical to the production line. The team combined their single-cell data with genetic information from 16,000 dairy cattle, uncovering new genetic markers for traits like fatty acid composition. These discoveries offer a treasure trove for breeders aiming to enhance milk quality and sustainability. But here's the twist. While the study celebrates scientific progress, it also raises ethical questions. Could manipulating these genetic pathways lead to unintended consequences, such as health issues in cattle or ecological imbalances? Professor Sun emphasizes that this work fills a critical gap in bovine biology, providing a foundation for future research in animal breeding and human medicine. The team's analytical framework, he notes, could even serve as a blueprint for studying other livestock species. Yet, as with all powerful technologies, the real challenge lies in how we choose to use these insights. What do you think? Should we prioritize efficiency in dairy farming, or should we tread carefully to preserve natural genetic diversity? Share your thoughts in the comments below. This is a debate that's far from over.